Hello everyone. What's actually the most expensive thing you own? Your phone, your crypto? Honestly, not even close. It's something you are carrying right now, locked behind the most high-tech security system in this planet, your genetic code. Welcome back to Illusion Hashtag. Today we are not hitting a bank. We are pulling off the ultimate biological heist. Forget about diamonds, golds, or anything. We are cracking the seal to steal the blueprint of life itself, the DNA heist. Let's get it. All right, team, focus up. Here's the brief. Our target 100% pure intact DNA, but it's not going to be a walking in the park. We are up against thick cellular walls. Think of them as reinforced concrete and aggressive enzyme that acts like laser and guards ready to shred our loot the second we break in. And so why we are even planning this haste? Because in the wrong hands, your DNA is not just a science project. It's the ultimate master key. We are talking about the entire identity, your health secrets, and Loki, maybe even the passwords of to your life in the future. This is not just data, it's the blueprint of you. The mission is simple: breach the perimeter, grab the code, and get out without destroying the evidence. Mission starts now. Now, every haste needs a tailored strategy. We are not just hitting a random lab, we are hitting data rich environments like ancestry sites. At first, we need to scope the target. Are we hunting the massive chromosomal DNA or small circular plasmids? Every organisms, from bacteria to fungi to human cells, deploy widely different defense mechanism. We have to know exactly what we are looking up against before we make a move. Whether we are extracting from an entire organ or just isolated from single cells, our methods depends on the loot we want, the molecular weight, the purity, and how much time we have before the gut catches. Pick the wrong strategy and the whole mission goes sideways. Okay, we have scoped the target. Now it's time to actually break into the vault. In the bio world, we call these as a cell lysis. Think of it as an ultimate smash and grab. We have two main ways to breach the perimeter. First up, mechanical disruption. This is a brute force method, grinding, ultrasonications, or using osmotic pressure to literally shatter the walls. It's loud, it's messy, but it gets the job done. And if we need to be a bit more sophisticated, we go with chemical lysis. Think of this as an acid that melts the lock. We use detergents, lytic enzymes, or alkali treatments to dissolve those defenses without breaking the suit. Now we're inside the vault, but the alarm is blaring. Just breaking the cell is not enough. We have to deal with the high security cellular guards waiting to ruin our hull. If our target has extra heavy proteins, armor, or aggressive RNA interface, we bring the specialist. First, we deploy the proteases. Think about enzymes like proteinase K as our heavy hitters. They ruthlessly hunt down the degraded structural proteins before they can tangle up our DNA. Next, we release the RNAs. These are the precision hacker. They explicitly digest any unwanted RNA molecule, clearing the field so that our target DNA stays pristine, isolated, and ready to transport. We are not actually leaving any witness behind. It's time to clean up the scene and secure the loot. Now we have cracked the vault and neutralized the guards. Now comes the most critical part, the gateway. We have the loot, but it still mixed with all that silver trash. To get the DNA out clean, we use two primary gateway vehicles. First, solution-based method. This is the classical chemistry play. We mixed our cell extract with specific chemical solutions to force a phase separation. It's like a high-speed chase where the DNA jumps into the different guard to lose the trail of the debris. Or we can go with the solid phase method. Think of this as a high-tech physical trap. We use silica columns or magnetic beads that act as a tractor beam, grabbing the DNA and holding it tight while everything else just washed away. Once the debris is cleared, we release the DNA, and just like that, the haste is complete, pure, intact, and ready for the next level. And as we already said, the choice of this extraction is actually depends on the target DNA, source organism, desired purity, and the extraction time. Let's get into the exact methods now. If we need maximum result, we call organic extraction. Think of this as a sledgehammer of the haste. It used organic solvents to ruthlessly kick cellular junk out of the equation. And the stats is like high yield, high purity, but watch out, its toxicity is high. This is not an amateur. Use it, grab the loot and get out. Now, to pull off this heavy-duty extraction, we deploy a specific chemical crew, phenol or point man. He dissociates the protein stubbornly bound to our DNA. Chloroform, the cleaner. He denatures the remaining proteins and lipids to clear the path. And isomer alcohol, the specialist. He keeps the reaction stable and prevent excessive foaming so we can stay stealthy. One wrong move and the mission is blown. Precision is everything here. Now, after this mixing the things, we force the separation. After a high-speed spin in the centrifuge, the loot settles into three distinct layers. The top, the aqueous phase. Basically, this is our hydrophilic safe haven. This is where the DNA and RNA is hiding. Middle is an interface, the thin dividing line, and bottom is the organic phase, the dumping ground for the denatured protein and lipids. Now let's talk about strategy for this final separation. To get out the loot, we deploy our chemical bouncers to force every component into the specific layer based on its vibe. Actually, it's their chemical properties. Here's the breakdown is like phenol is our point man, and while the phenol is busy, chloroform comes in as a cleaner. He denatures the remaining proteins and lipids, trapping them at the bottom so they can't mess with the prize. And it's liver aqueous phase, the top layer, the hydrophilic safe even. That's where our target DNA and RNA is chilling. It's all about like forcing the junk out so the coat can shine. Now, after all those things, now it's time for the final stage, the cleanup. Our target DNA is currently trapped on the top aqueous layer, so we treat it with ethanol. This caused the DNA to crash out of the solution, forming a pellet at the bottom after one last spin. The liquid is completely discarded. The price it's secured. And one pro tip is like we added isomer alcohol to the mixture earlier for a reason. It's the specialists that prevent disastrous foaming during the reaction, keeping our mission smooth and stealthy. And here's the chemistry recap for the final last time, which is like a phenol dissociates the protein crew from our DNA, chloroform denatures the remaining junk so it can't follow us, and isomer alcohol prevents the reaction from foaming over and blowing our cover. And the result, the DNA RNA stays safe in the stop aqueous layer phase, while the cellular garbage stays trapped in the organic phase and interface, and the loot is finally secured. Next up, we have the salt-powered magnet. Think of this as the high-tech tractor beam of our Heist. Is it like we need to switch it on by drop a high-salt alkaline environment? This forces our negatively charged DNA to bind tight to the positively charged silica, basically locking the loot in place while washing away the trash. And after that, once we are ready to leave, we need to switch it off. We wash it with the nucleus-free water or the elution buffer like 3CDTA. Thus, we let the DNA go and we are officially in the clear mode. And deep dive into the silica trap, this method basically relies on the high binding affinity and the specific environmental triggers. And one pro lab tip is like you must have guanidinium thiocyanate, that is GITs in the mix. It is the glue required for the DNA to bind to the silica in the first place. And here's a quick recap. The first thing is that the trap is basically based on the environment, alkaline high salt, and the mechanism is like negatively charged DNA will bind to the positively charged silica. And once this is done, the washing with the elution buffer like 3CDTA. And the last step in our arsenal is the zero spin superhero. This is the ultimate stealth extraction method. The tech we use the paramagnetic beads coated with the DNA binding functional groups. They find the loot and hold it tight. And the best part about it, no centrifuge required. We use magnets to move the beads instead of high speed spinning. This removes the stress and shearing forces that can shred the sensory molecule. Your target stays 100% intact, means haste completed with zero drama. Let's look at a magnetic pull in action. The first thing is the trap and then the clean. Here you can see the final four-step execution. First is bind, DNA hook onto the beads selectively. Now applying. This it is like click that external magnetic field into the place, then pull. Watch the beads hug to the tube's wall while the junk stays in the solution and then it's released. Watch the trash and unlock your pure DNA and your haste is complete. The final asset of the toolkit, the electrostatic trap. This is the heavy duty tech we use for high purity mission profiles. Here's that execution part is like we use matrix, that is DEAE cellulose, which is basically positively charged molecule that can act like a high voltage magnet for our load. And the target is our negatively charged DNA backbone, locks onto that matrix instantly. And the protocol is very simple. We flush the contaminant with a low salt wash, then drop a high salt buffer to force the pure DNA to let go. And though you are getting the Li purity, but the catch is you need to dissolve the price before it's ready for the next level. Think of this anion exchange as a chemical velcro. It's not just a general affinity, it's a spatial specific interactions. And the thing is the grip for which you are using basically this high positively charged DA resin, the release by flushing with a high salt buffer, and the score is the extremely pure DNA. But yes, before using it, we need to dissolve the thing. And now the boss level tool, the plant killer. We save these for the toughest target, plant samples packed with tricky acidic polysaccharides. And the strategy, the reagent we used, that is CTAB extraction buffer at an alkaline pH. And the trap is at low ionic strength, both nucleic acid and those anion polysaccharides precipitate out together. And then we heat it with a high salt concentration. These solubilize our DNA load, leaving the polysaccharide trapped in the precipitate, and that is a success of this mission. Now, why the specialized gear? Because the plants will deploy thick walls and complex sugar as biological firewall. Standard method fails here. So the CTAV method is a specific countermeasure. By using cetyl methyl ammonium bromide, we force the enemy's own defense to precipitate out, leaving our target DNA pure, isolated, and ready for extraction. Now for the final switch up to secure the score, once the initial CTAB nucleic acid complex formed, we heat it with a high salt concentration. Here's the wins like the DNA loop becomes solubilized in the high salt, while the acidic polysaccharide can't handle the pressure and remain behind as a precipitate. We then wash the purified DNA using our trusted organic solvents to, that is phenol, chloroform, and isoamyl alcohol, to leave the scene clean and firewall is down. And here the final objective, the sniper rifle. Sometimes we are not after the whole world, just a specific high value asset we are looking for. We are talking about isolating tiny circular plasmid DNA from the massive bacterial chromosomes. And the challenge is like during the extraction breach, the large chromosomal DNA often breaks into messy linear fragments. But our target, the plasmid, still locked in the covalently closed supercoiled circular form. The separation relies entirely on physical size and conformation. It's a precision operation to pick out the small circles from the massive tangle of linear debris. Now, to separate our high-value plasmids from the chromosomal mass, we force a total structural crisis. And the move is like first we attack by dropping a high concentration sodium hydroxide solution into the mix to renature every strand of the DNA present there. And because of this, the linear thing, the massive chromosomal fragments completely collapse as they lose their grip and their strands stripped apart from this tangled mass. But for the plasmid, they are topologically constrained. They denature, but because they are supercoiled structure, they physically cannot drift apart. They stay locked together while the rest of the world collapse. Tactical precision at its peak here. Now it is a time for final counter move. To secure the loot, we rapidly introduce a neutralization buffer of potassium acetate. This triggered a massive tangling event that only one side survives. First, our topologically constrained plasmid DNA instantly snaps back. It reannul perfectly and stay cleanly solubilized, ready for the pickup. And the trap is like the long denatured chromosomal DNA attempts to reannul, but it panics because their strands floated far apart. They cross-link incorrectly, reaggregated into massive tangled insoluble network. The result is like one final spin in the centrifuge easily plates this heavy tangled mass to the bottom. This leaves our pure plasmid DNA suspended in the supernatant, alone and secured. And by this way, the mission is fully accomplished. And here, there is a quick recap of this snapback technique, the attack, the linear collapse, and the circular win. Now, if we need even higher precision, we deploy the elegant density sorter using cesium chloride and Ethereum bromide, that is ETBR. The ETBR is an intercalating dye that violently forces its way between the base pairs, causing the double helix to unwind. And the larger linear DNA has free ends and unwinds easily, binding the massive amounts of ETBR and dropping its density significantly. But the supercoiled plasmid have no free ends and are too topologically strained to unwind much. They absorb way less ETBR, keeping their density high and forming a distinct heavier band in the centrifuge. And that is a pure precision and pure code. And here you can get a brief idea of the position of various bands in the cesium chloride density gradient method. And finally, the master lab toolkit defined by the precision. This is all the techniques we have discussed today and their specific use for the things. First, we started with the organic extraction method. Then the silica for the high salt solid trap. We then proceed with the magnetic beads method. For then anion exchange for CTAB for the plant thing, and for plasmid alkaline denaturation and cesium chloride density gradient method. So, what's the move? Is a biological haste something we should actually be worried about in 2026, or is it still just pure science fiction? Let me know in the comments if you think we are ready for the level of this level of tech. If you enjoy the breakdown of this ultimate molecular bridge, hit the like button and subscribe to Illusion hashtag for more deep dive into the shadows of the tech and mystery. We have fully unlocked the vault of DNA extraction. Mission accomplished. I am out. Bye.